everyone, Dr. Yu here. Today I want to talk to you a little bit about stains. So when we actually get the stains into the lab, they come in a powder form. And we take that powder and we dilute it into a solvent. That solvent is either going to be water or ethanol. Now that powder is actually what's called chromogen. Chromogen is made up of two different parts. We have chromophores and we have oxychromes. So the chromophores are going to be the colored portion of your actual stain, and you can have one chromophore or you can have multiple chromophores. Usually if there's multiple, it intensifies that stain color. And then we have these oxychromes. Now the oxychromes are actually going to be the charged portion of the stain. Now when we look at this, we're going to be working with bacterial cells, and bacterial cells actually have an inherent charge associated with them. They're negatively charged. So it's important that our dye have a charge associated with them so that there can be interaction with the bacterial cells. Now, when we look at stains, we see that there's two general forms of stains. We have basic stains and acidic stains. And before I get into talking to you how that actually works and what that interaction looks like with the bacterial cells, I do want to back up and talk to you a little bit about pH. Don't panic, you're not going to be having to calculate any pH of these staining solutions. I just kind of want to walk you through this so that you get a better understanding for why these are named the way they are. Okay, so when we determine pH of a solution, we use this formula. So the formula is negative log of the hydrogen ion concentration. So pH is based off of the amount of hydrogen ions, or H pluses, that are in a solution. So if you increase the number of hydrogen ions in a solution, this actually decreases the pH. So there's an inverse relationship there. You go up with the hydrogen ions, you go down with the pH. When you go down with the pH, you become more acidic. Now, the opposite is true. We have a decrease of hydrogen ions. That leads to an increase in pH, and that causes the solution to become basic. So that's important to note for this next part where we talk about these actual stains and how they work. So our basic stain, it's going to have that oxychrome and chromophore, and when you mix it together with your solvent, what this actually does is it decreases the amount of hydrogen ions that are in that solution. So we just learned that if you decrease, then your solution becomes more basic. So that's why this is referred to as a basic stain. Now, these hydrogen ions don't just disappear. We actually see that the dye itself will attract the hydrogen ions from the actual solution. So now our dye or stain takes on a net positive charge. And this is important because when you mix it with a bacterial cell, these bacterial cells have a net negative charge associated with them. And what do opposites do? They attract. So we actually see that the dye will be attracted to the negative charge of the bacterial cell. And because of that, the dye will stick to the bacterial cell, and the bacterial cell will take on the color of that particular dye for that stain. Now for our acidic stains, we see a little bit different scenario going on. Here we have the dye, you add it to your solvent, and we actually see that the dye then gives up some hydrogen ions into that solution. So now we have more hydrogen ions floating around in the solution. That increases the hydrogen ions in the solution, and we know you increase, you lower the pH, it becomes more acidic, hence the name acidic stain. Now because the dye has given up, or the oxychrome has given up some of those hydrogen ions, it now has taken on a negative charge because it's given up some positive charges. So now we take our acidic stain, we mix it with our bacterial cell, and now we have two of the same charges. So what's going to happen there? Well, whenever you have two of the same charges, they repel. So we see that the stain will actually be repelled from the bacterial cell. So our bacterial cell will remain a clear color, and the background will actually take on the color of the stain. This is why it's important when you do these types of stains, you usually don't do a final rinse because you don't want to rinse away the stain from the actual background. Now here we're staining the background and not our actual cell, so this is kind of like a negative, so this is referred to as a negative stain. And here, when we are actually staining what we want to see, we're staining the bacterial cell, we refer to this as a positive stain. So that, in a nutshell, is how our basic and acidic stains work. If you have any questions, don't hesitate to reach out to your instructor.